All right, so this is the second talk in our cell cycle videos, and at the end of the last one I forgot to cover the cell division by binary fission. So this is how your prokaryotic organisms undergo mitosis or cell division. And so as you recall, our prokaryotic cells have circular DNA, so they have one origin of replication where they start the replication of their DNA. And so once the replication of the DNA starts, you're getting the semi-conservative replication. So each old strand is getting a new strand made on top of it. And so now, once you get enough replicated, you can move the two new origins because you're having two new complete copies of the DNA. They kind of move to opposite poles of the cell. As they move, the cell continues to elongate, continues to make more ribosome, cytosol, anything that the cell is going to need to make two new copies. So it continues to elongate, and then once it reaches a certain size and replication of the DNA finishes, then it pinches in half, has tubulin-like proteins that help to pinch the cell in half and create two new daughter cells via binary fission is how prokaryotic replication takes place. All right, so how do we control the cell cycle, and what happens when cell cycle control is lost? So our cell cycle has three main checkpoints. We have our G1 checkpoint here in G1 of interphase. We have the G2 checkpoint at the end of interphase, and then we have a M checkpoint right here at metaphase before we move into anaphase. And so the G1 checkpoint is actually the most critical. It's sometimes called the restriction point. And so the restriction point is where it decides, do I have enough things? Do I have growth factors? Am I nutritionally healthy enough to go? Am I big enough? Do I have enough nucleotides? Do I have enough enzymes? Do I have everything I need to complete my cell cycle? And if it's yes, then it's going to move into the cell cycle. If it's not, it's going to move out into a phase called G0. And that's where it's a resting state. And so it'll go there and rest and try to regain energy so that it can pass this G1 checkpoint. And so things that regulate these are called our cyclin-dependent kinases. And the cyclin-dependent kinases are activated when a cyclin molecule binds to them. And you'll get to see that on our next slide. And so the CDK and cyclin that regulate the G1 checkpoint are CDK2 and cyclin E. And if everything's good and it can go, then the barrier comes down and it moves on into S phase where we do DNA replication, G2 when we finish preparing for the cell cycle. And here we have another gate that does a checkpoint. And so it's going to look, was our DNA fully replicated? Is our DNA intact? Does it have integrity? Is it good? And if it does, then it'll go ahead and move into our mitosis or our cellular division. G2 is regulated by CDK1 and cyclin B. And once it moves into mitosis, once we're at metaphase, we have another checkpoint, M2, and it checks to make sure that attachment of all the kinetochores has been achieved so that every sister chromatid is attached to a mitotic spindle. Because we need to make sure everybody's attached or somebody will get left behind and then you won't get two complete copies of that in each cell. And so if everything's good, everything's happy, every kinetochore has attached to a mitotic spindle, then this activates an enzyme called separase, which allows sister chromatids to separate. And this is regulated by APCC, sometimes called cyclosome. And so again, another illustration in your text, how if cells are not nutritionally ready at the G1 checkpoint, they're pushed into G0. But if everything's good, the gate is lowered and they're allowed to proceed. And this is that restriction point. It's a very critical checkpoint. And the other critical one is the M checkpoint. G2 is important, but it seems that G1 and M are two of the most critical. And again, this is making sure that the mitotic spindle is hooked to a kinetochore of each sister chromatid so that they can each be pulled to opposite poles in anaphase so that every new daughter cell gets a complete copy of the genetic material. And so again, everything connected, everybody happy, gate is lowered, can proceed through the cell cycle. And those cyclin-dependent kinases I told you about earlier, remember anything that has ASE, that's an enzyme. So cyclin-dependent kinases are a type of enzyme. And so they are always there. And another thing that regulates our cyclin-dependent kinases is phosphorylation. And so certain spots, if it's phosphorylated here, may inhibit it, but if it's phosphorylated over here, it may activate it. But also for activation, a cyclin needs to bind into that activation site to activate the cyclin-dependent kinases, and then those guys push the cells through those restriction points into the other phases of the cell cycle. But again, remember, if they don't meet that restriction point in the G1 checkpoint, they go out and rest in G0 until they can recoup and get enough energy to move through the cell cycle. And here's those cyclins. So those cyclins are turned on and off as the cell moves through the cell cycle. So you have those cells, those genes, coding for those proteins, turned on and cycling throughout the cell cycle. So you have different cyclins at different times for those different checkpoints to help you move through. And you can watch the animation on this with the Amoeba Sisters at this URL. Other things that regulate cell cycle are density-dependent inhibitions. And so when cells are growing, and when they touch each other, they have receptors on their cell surfaces, and when they touch, they're like, oh, we're neighbors. We don't need to grow anymore. Let's stop. And so they stop when they touch. They like to hold hands. Now, if you took a couple guys out and opened up some space, then that inhibition would be taken out and they could grow again. And so then you would go back until you touched hands again. Everybody's holding hands. And then they would stop. Now, when you lose this density-dependent inhibition, this is when you start getting cancer cells. And you can see how this was a nice single layer of cells. Over here, when they lose that contact inhibition, that density-dependent inhibition, then they start to grow on top of each other and grow uncontrolled. Other things that can cause issues in our cells are when we have some genes. We have two types of genes that are going to affect cell growth, help regulate at those checkpoints, and those are tumor suppressor genes and proto-oncagenes. And here with our tumor suppressor genes, you can see if both copies of our tumor suppressor gene are happy and normal, we have controls. We can slow the car down. We can stop the car so that we don't run into a wall. If you mutate one of the tumor suppressor genes, because remember all of our cells have homologous chromosomes that code for the same type of genes on each one, then you can almost stop and you can still control it because the good gene still makes enough. But if you mutate both, you're just going to slam right into the wall and grow uncontrolled. You're uncontrolled. And so this is a unique feature with tumor suppressors, is that you have to mutate both copies for complete loss of control. There are some instances with some of our stronger tumor suppressor genes like P53 where they only have to have one copy mutated, but the majority of our tumor suppressor mutations are going to have both copies mutated for uncontrolled cell growth. And what is uncontrolled cell growth? It is cancer. And so this is an illustration of how tumor suppressors control the cell cycle, but as they get mutated, they start to lose control. And when you knock out both, there is no control over our cell cycle anymore. Examples of those are P53, retinoblastoma protein, APC, and the BRCA genes. Another type of regulating gene, the second most common, is our proto-oncagenes. And so a proto-oncagene is a gene that exists in our cells at all times. And so the proto-oncagene, when it is not mutated, the proto-oncagene functions normally and you have a normal cell. But when this proto-oncagene is mutated by a cancer-causing agent, it turns into an oncagene. And when you have an activated oncagene, then you have a cancer cell. And so you can think of this is the gas pedal. So you're pushing the gas pedal, you tell the cell to go, you take your foot off the gas pedal, the cell stops. And when you mutate it, the gas pedal is stuck. It can no longer be stopped. And so oncogenes are what cause the cancer. When a proto-oncagene is mutated, it loses the ability to undepress the brake pedal or undepress the gas pedal. And so you lead to cancer with a mutated proto-oncagene also. And only one copy of a proto-oncagene actually has to be mutated for the uncontrolled division to take place. Several examples of oncogenes are growth factor receptors. So growth factors are things that push cells into cell cycle division, help to enhance cells to grow when you need them. And so if you have a mutation in the receptor, then it's turned on all the time. So it thinks that a growth factor is bound to it at all times, so it just grows under control, uncontrolled. Other examples of proto-oncogenes are RAS, MYC, SERC, telomerase. And so we'll talk about telomerase when we get to DNA replication. This is actually thought to be, if you could ever harness telomerase, you could have the fountain of youth. And here's a real interesting illustration. So the size of the gene, P53, tells you how much, the larger the gene name, the more frequent that gene causes these types of cancers. So these are brain cancers, other types of ear, throat, mouth cancers, lung, breast, skin, liver, bone, stomach, pancreatic, the intestinal cancers, colon cancers. And so you can see who's predominant. P53 is almost everywhere. P53 actually causes about 50% of all cancers. Retinoblastoma, about 40%. RAS, 20 to 30%. Growth factor receptors are in a lot of breast cancers. SERC. And you can see the BRCA2 and BRCA1. BRCA1 and BRCA2 are so well known because they're inherited. So you can inherit that mutation. A lot of these other mutations occur for other reasons. They're not necessarily inherited. But I thought this was a really neat illustration of the different defects in our genes that cause these type of cancers and how frequently they are responsible for these types of cancers. And so another controlling mechanism, so we've looked at density dependence, we've looked at tumor suppressors, we've looked at proto-oncogenes. These are all things that when they are mutated or that control is lost, lead to cancer. Another one is this anchorage dependence. So when a cell loses its anchorage dependence, usually a cell must be attached to something like your connective tissue or your basement membrane or something that it grows on. It likes to be attached to something. It likes to be held down. But when it loses this anchorage dependence, it can metastasize and move. So many things cause tumors, and you have uncontrolled cell death, and then when they lose that anchorage dependence is when they can actually break free and become malignant, meaning that they can move through the circulatory system or the lymph system and spread throughout the body. But cancer cells, because of the mutations, usually end up exhibiting cancer cells lose their density dependence inhibition and their anchorage dependence because of the mutations in our various genes. All right, so that's it. I hope you guys enjoyed the discussion on our cell cycle and the controls of our cell cycle. And if you have questions, be sure to reach out to your instructor because they are there to help. Have a great day.